Okay, it's done. Okay, so you're going to do single molecule FRET. And the question is, what is this? Oh, by the way, has anybody done single molecule FRET in this course? No. Okay, good, because we've kind of gotten screwed up. Okay, so do I need to remind you of FRET? No. Okay. I'm going to remind you anyways. It'll just take a second. You have a donor, you have an acceptor, you excite the donor, it can either fluoresce or it can transfer energy to the acceptor, at which point the acceptor will fluoresce. It turns out it's what's called a dipole-dipole interaction, meaning the energy transfer goes as 1 over r to the 6. So when they're very close, it's a lot. When they're very far, basically the donor doesn't know about the acceptor. And you see a curve like this. The donor acceptor is close by. That means the donor isn't spending much of its time fluorescing because it's spending its time transferring to the acceptor, which has a lot of fluorescence. At a distance r naught, about half of the donor's energy goes into fluoresce, and about half of it goes into energy transfer. Okay, now, so that wasn't that painful. Now the idea is to do it at the ultimate sensitivity. The ultimate sensitivity is a single donor and a single acceptor. And it's, in terms of FRET, it was in 1996 that actually Tech Chip was the graduate student. He was the first author on the paper showing that from a single donor to a single acceptor, you could get energy transfer. Okay, so what we're going to do is, you are going to do it you're going to take a piece of DNA. And actually, the DNA is a little bit complicated. It forms what's called a four-way junction. In particular, it has 4 pieces of DNA, and it wraps up structure that looks like this, and you have A, B, C, and D. And what you're going to do is you're going to put a dye on B and another dye on A, B and A. And on C, over there, you're going to use a small molecule called biotin. Biotin binds very well to what's called streptavidin, which is another dye. And what you have your cover slip. Yeah, can everybody see this, or do you want more like, okay. You have your cover slip, and you have a protein called streptavidin. Streptavidin will bind to a molecule which is called biotin. Biotin is a really little molecule. It's even smaller. It's about the same size as a fluorophore. And the binding of these is impossibly good. It's, in fact, it isn't a covalent bond, but it's the strongest non-covalent bond that there is. So what you do is you have a DNA that has biotin on the end. That's shown right in C, and basically it'll bind to other DNA molecules. Such that you have two double helices, essentially. And it turns out that actually it can be in two conformations, such that, you know, D and C, B and A, where the distance between, where are we doing it? Yeah. We put the acceptor, which is CY5, the acceptor, right here, and on I don't know, so something like here, you put, which is your donor. And it turns out that this will undergo a conformation such that, you know, I don't know, D and B and C and A will sort of do a flip. And you'll actually go and measure it. So for example, let's say you measure this. In green is the donor fluorescence, and in red is the acceptor fluorescence. Tell me, I want you to do this on your own, or without me. Tell me what's happening with the donor and its transferring energy to the acceptor. Is it a lot or little? And in fact, you can get the FRET by looking at the relative amount of donor and acceptor, right? If the donor and the acceptor are far away from each other, if the donor and acceptor are far away from each other, how much energy transfer will you get? Very little. So the donor fluorescence will be big or little? Large. Okay. Now if it undergoes some conformation such that the donor and acceptor are close by, how much is the donor fluorescing? Big or little? What's that?
Oh, the donor, and how much?
Not much. And how much is the acceptor fluorescing? Quite a bit. Okay, so good. Go tell me about where they're close and where there's far, and you want to get out some sort of time scale for how the donor and acceptor, how they're close and when they're far. So, and why is there all these wiggles? Okay, so go ahead. So.
Spaces of time are they transforming, and what spaces of time?
So Lydia, make sure all three of you are involved.
Especially now it's the disabled are very close for the acceptor.
So something fluorescence as far as time scale.
Not only the fluorescence.
Yeah. So the time, so it's let's just say the amount of time it spends in the other stage looks like it varies a lot.
I mean, it should fluctuate.
The amount of time it takes to go between the two stages also.
Yeah, they kind of just fluctuate. Turn out and sit it. I don't know what it's like here. It's a four-way part. No, it's this four-way junction. If you look on the board.
Yeah. There's a side 3 and a side 5. Is that what it's like when you
It looks like it's
because
more
you know,
time.
like transfer RNA, that's what I automatically think.
Is that transfer
No, I don't think
on the upper half.
Well, it's just how sometimes DNA forms.
So
Yeah.
It has this little
This is four-way junction.
maybe this is like the
Is it four-way junction, holiday junction, or those are different, right? Holiday's two strands.
I'm not actually sure. Yeah, I don't know if they form a two-way or three-way or four-way junction. I'm not sure. So you all talked out? Don't try too hard. Okay, go ahead. Go ahead. Come on up. All right. Go ahead. Tell me what's going on.
Tell you what's going on. That's a tall task. All right. To start out, me and Greg talked about how we looked at this and how when the red line is higher, that means there is basically they're closer together because you're getting more fluorescence from the acceptor. And then places like here, you're getting more fluorescence from the donor, so they're further apart. And then beyond to that, I was like the inherent randomness of what's going on here. I was struggling trying to find an explanation for that would be useful.
Looking at the bottom graph, what does that tell you?
So,
And what is the bottom graph?
so this is, I'm guessing this is efficiency of energy transfer, like the value you get from the FRET.
Mm-hmm.
So when it's this area, there's a lot of FRET going on. It's going to be, the donor and acceptor are going to be pretty close together. Here, they're going to be pretty far apart. Looks like it spends more time being close together than far apart. On the top right is side 3 and side 5 is side 3 donor and side 5 acceptor. Are those the things?
Okay. What about it? Is that a two-state system? That is the FRET either 0 or high, in this case 0.8, or is it actually a three-state system?
Well, there's three dotted lines.
That's a hint, yes.
So I'm guessing it's 3-state.
Okay. Let's say the dotted lines weren't there.
Okay.
What would you say then?
Yeah, there's definitely like 3 distinct areas that like
Yeah.
to go to interactions.
Okay, so apparently this thing isn't quite what I said, that there are actually 3 conformations. One where the donor and acceptor are very far apart, one where they're quite close, and one where they're intermediate. Okay, so It's good enough.
But why does it transition between 3 and 2 much faster than the transition between 3 and 1?
Ah,
I think that the furthest away distance would be D and B, and then the closer together is if the side view was what D is. And then if it's close together, it's like it's shown in the image. And I think the way it's shown in the image is like these two shoulders just come together. Something like that.
something like that. Now you're interpretation. And in fact, one thing that he found was that going from E3 to E2 is pretty common, pretty fast, and going from E3 to E1 is fairly slow. And this will give you a hint as to what the states are. Yeah.
So the reason we call E2 the third state, other than just fluctuation was the high fractal state, is that it's just beyond the level of square root of the
That's a good question. Why do we say that this is sort of a distinct signal rather than, let's say, you have some fluctuations in the amount of photons coming out? Well, this is FRET. This is the intensities. So what we're finding is that there is this third state where the donor has gone down, and at the same time, the acceptor has gone up. Right. And so, so that tells you that actually it's something that's changing that is changing FRET. So actually, how do you go from here to here? How do you make this graph?
Well, you compare the intensities and the iteration.
Good. So, okay, you compare. So what do you do?
Acceptor over donor intensity.
What is it? What? This is. And this E, this is the FRET?
Almost. Yeah, basically.
Okay, but. What do you say basically?
We still need quantum field.
We still need quantum yields. Because actually, what is the real formula? Quantum yield of A over A over quantum yield of A plus D over quantum yield of D. This graph is just this. So what do you do about the quantum yield of A and the quantum yield of D? Either you just say they're equal, or more often what you do is you go and measure the quantum yield of D and the quantum yield of A, and it's very difficult to measure it actually on the sample that you're looking at, because remember, the sample that you're looking at is a single molecule. So in general, it doesn't last very long. In what sense does it not last long? What? Yeah, photobleaching. What happens is the acceptor, the donor, photobleaches. So if you want to do a quantum yield measurement on the acceptor, then quantum yield on the donor, and then you do a FRET experiment, the chances are it'll be dead before that. So you generally look at a whole nother sample. And you get some ideas for what QA and QD. And okay. What happened there? You guys are really good at reading my slides. Okay, what is photoblinking?
Just by chance at one point so
The acceptor is just off.
Acceptor is just off. And yeah, you get all sorts of weird behavior. The acceptor is just off. And interesting, it's just off. What does that mean? Does that mean it's off in terms of it's not emitting, or it's also not accepting, or what? Which?
It touches goes all the way to zero.
Well, okay, this might, we'll get to that. Let's say this is about 0. And yes, it looks in general to me, this looks like this, although this might be a little different. So again, is the acceptor, is it not fluorescing, or is it not absorbing, or is it both?
oh, it's not a simple absorbing.
It's not absorbing. Why?
We still see the donor emission.
You still see the donor emission, and therefore?
Donor photons are free to move. They're not being absorbed and taken out of the emission spectrum.
That's right. And so this guy is not only non-fluorescing, it's not fluorescing because it's not even absorbing. So the way you know that is because you look at the correlation. And it looks very similar over here. Now, so what has happened is the acceptor just goes into some unknown state where it's not accepting and not emitting. And then apparently it comes out. And the same thing is here, and all of a sudden it comes out, and what is the acceptor doing it in terms of absorbing and emitting? It's starting to absorb and to fluoresce, and because it's absorbing, the donor is decreasing. Okay? Okay, and that's basically the story. Yes?
Well, like, as what he asked,
Ooh.
would that count as a photoblinking time lapse?
Well, this would certainly be counted as a photoblinking time, and this would certainly be counted, though that whether you count them up in terms of your statistics, in general, I would say you probably ignore that, though it's somewhat up to you. The reason that I would say you ignore that is you don't really know, like over here, was it half the time on and then half the time off? So in fact, you shouldn't count this as 0.5. Maybe if it was on a little bit longer, it would have gone to 1.
But why would you count this photolinking and not just the difference in confirmation?
Well, because this, I would say, is been photoblinking, and this is photoblinking. Here, it comes up to about 0.5. I mean, both the acceptor goes up and the donor comes down, indicating that there is some sort of FRET happening. Though what I'm saying is the exact amount of FRET you don't really know because the molecule maybe wasn't fully turned on, that sort of thing. Okay? Okay. Now, what I said is you have your cover slip, you have your streptavidin, and then your biotin DNA. And you typically shine a laser at it. Say, you shine a laser at it. Who's done the Fiona? So you learned during your Fiona a little bit about TIR, total internal reflection? Yes? Okay. So for the other people, you're about to learn it. And for those of you who already know it, you might learn a little bit anyways. Okay. What you have is if you just come up and shine and shine the light, and let's say you put your cover slip right on the water surface, you go there, and it basically passes right through the water. And so if you have if you have some of the DNA that's not fixed to the surface and it's coming up, they'll fluoresce. And that's a pain in the ass, right? So in general, what you want to do is you want to limit your excitation range only to those nearby. And it turns out that this is a very standard tool that not only will you do in single molecule FRET, but also you do with Fiona. So how do you do it? Well, essentially what you do is you do this. You change the angle at which, why don't we shut off the lights? Everybody see? So this is your laser coming in at a certain angle, and you change the angle. And if you have objects that are above your cover slip, they'll be excited, right? They're still excited. And oh, look, what's happening? as you bring it out, you're not getting anything up there anymore, right? What determines that? What? The index of refraction, that's right. But so, how do you do this a little more quantitative? What is the law governing this? Yeah. Okay. So Snell's law no. So what? Oh yeah, go ahead. Turn them on. We'll shut them off in a second. What is Snell's law? This is theta 1 going through some material which has an index of refraction n1, and this is theta 2 going with an index of refraction of n2. Okay, so what you see, if theta 1 is equal to 0, sine of...
I think theta 2 should be on top.
Good. Now let me see if you're correct. So does this make sense? N1 sine theta 1. So if theta 1 is equal to 0, in this diagram, it's coming straight up. And so the sine of 0 is 0. 0 is equal to n2 times sine theta 2. Therefore, theta 2 must be equal to 0 also. Okay, that's good. And now, let's say it's at 45 degrees. Sine of 45 degrees is what? Yeah, okay. N2, and then sine theta 2. So sine theta 2 is equal to n1 over n2 times the square root of 2. Okay, that seems right. Okay, now, what about, so can I have the lights? Okay, so now you go, what happens when theta 2 is equal to 90 degrees? What's sine of 90 degrees? 1. And so, can I have the lights on again for a second? So theta 2 is equal to 90 degrees, so that's 1. So n1 is that right? Okay, so sine of theta 1 is n2 over n1. n2 in this case, N2 is the air. So that's an index of 1. N2 is water. N1, I'm sorry, is water. Do you know what the index of refraction of water is? 1.3. Or 1.33. Okay, so somebody who has a calculator, figure out what is theta 1. Somebody must have a calculator. Now all of a sudden everybody goes for it.
175.
What's that?
175.
175?
0.75.
Yeah, that's sine theta 1. Now what about theta 1? 49 degrees? Okay. Okay. So when this guy, okay, can you? So here, when this guy, about 49 degrees, that means this guy is going, and it's just sort of, the energy is just penetrating. And it turns out, then you do it ever so slightly more, and you start to see some reflection over here, but that doesn't excite your molecules right on the surface. But it turns out, okay, can I have the, at that point when you're, just slightly, 49 degrees or slightly over, what actually happens is you get a wave. It's not really a wave. You get the light is totally internally reflected, which means that you have an exponentially decaying field right at the surface. The exponentially decaying length is typically about 50 or so nanometers. It's very small. And 50 nanometers is perfect for what we want, because in general this is about 50 nanometers or so. So what you do, and you will see, is you will go and you will adjust the angle of the laser coming into your sample, and you'll get it just to the point where all of a sudden you'll see fluorescence due to the total internal reflection. Okay, and now there is two ways that you can totally internally reflect it. One way, which is here, so here you're coming in with a laser, you're bouncing it off of a dichroic. What it means is the blue light, let's say you're using blue, gets reflected up into your objective. Though it's not right in the middle of your objective, it's coming in very at the edge. In that case, it comes in and it's about whatever was 49 degrees. It totally internally reflects and bounces over here. A little bit of the light gets to excite the sample. The sample fluoresces, in this case in red, and goes down into your CCD. That's one way of doing it. That's a fairly complicated way, although it's the most common, I would say. The other way that you can get to your 49 degrees is you have your sample here and you come in from up top. And instead of coming in like this, you come in like this, such that it's 49 degrees, comes like this, goes like that, and again, about 50 nanometers into the surface, you excite. So either one of these techniques. This is, for technical reasons, a little bit more common nowadays, although the prism type TIR was the most common way. Okay, and again, we've already gone through this, that you have lots of approximations here. In particular, you're missing the quantum yield of the donor and acceptor. Okay. Lastly, that you're actually going to do this not on simple DNA, but you're going to look at telomeric DNA. Does anybody know what the telomeres are? Yes?
So there are sequences in the DNA that repeat, and with each cell division, because the DNA has polarity, we have the 5' and the 3'. With each cell division, we lose a little bit of the telomeres. So as we age, we have shorter telomeres.
Where is this repetitive DNA?
They're at the end of the DNA sequence. Each chromosome.
At the end. Remember, you have 46 chromosomes, and so in a cell, when the cell or the housekeeping genes see an end of a DNA, it says, "Whoa, hold on. We got to do something here, because the DNA, maybe something came in and, sliced it up." In fact, often it causes the cell to die. So because you have not 46, but 46 times 2 ends of the DNA, the cell is very tricky. It hides the fact that there is a double-stranded break there. And it does it, in general, by using these particular sequences, which telomeric sequence, which is often TDA, GGG, and it folds itself such that it fools the rest of the cells. Now, what she was saying, Monica, was each time the cell divides, it needs to make a new DNA. And it does this, but not perfectly. It turns out it does this, but each time the telomere is a little bit shorter. And so after about 100 cell divisions, there's no longer any telomeres, and the cell dies, and you die. It's true. Now, you laugh. Now, maybe the cell wants to live. And occasionally, there are problems with this. It doesn't die. It, in fact, becomes immortal. And what do you call that? Cancer. You call it cancer. And that's what happens. Those cells tend to be immortal. They replicate and they replicate and they replicate, and pretty soon that you're one big cancer cell instead of regular. And about 90% of cancers are involved with the telomeres. Okay, and so you're going to study about the telomeres, and in particular, the sequence is generally called a G-Quadruplex DNA. And what you're going to do is you're going to, I don't remember actually where in the loops you're actually going to place the donor and acceptor. And then you're going to vary the amount of salt. So at 0 millimolar, at 0.1 millimolar, at 2 millimolar, at 10 millimolar, 100 millimolar, You have 100 millimolar over here. Then this decreases and this increases. At 2 millimolar, this decreases, this increases, and this increases. So apparently you have three states. Not coincidental that I showed you a three-state system. Yeah?
Is the salt competing with the magnesium that's happening? Can you add a higher concentration?
Sure. Unfortunately, I don't really know the biology of this. Yeah. Tick Chip, who was here but had decided to leave, would know all about this, or Sua Mwang, who is the first author of this, would certainly know.
I think it's hydrogen bonds.
Actually, the DNA is highly negatively charged. So maybe the salts interact with the negative part and they change the composition.
That's what I was thinking. I was thinking the potassium will displace magnesium. Magnesium, is it 2+, right?
Yeah.
Yeah.
Okay. And the last thing that I'll do is, you see, this sort of very wobbly diagram, similar to what we saw. You need a way to objectively analyze how fast, first, is it a two-state system or is it a three-state system? And if it, well, whatever it is how fast does it go here versus go here? And there is a method which is called hidden Markov method. We won't go into a lot of it, but this is the standard way of analyzing this kind of trajectory. And you get out, actually it's pretty amazing. Partly the question is what is hidden about it? Well, first, and what is Markov? And a Markov method is the transition rates between the states are independent of time. So one here, one here. It doesn't matter how long it's been in there. There's a certain rate, a certain number of times per second that it goes. And in here, it doesn't matter how long it's been here. There's a certain amount of rate that it goes. So that's pretty general. Why is it called hidden? Actually, there's sort of two reasons. One is there can be so much noise. This is what's really absolutely amazing about this. There can be so much noise because of photon noise or whatever that the signal is just lost. I'm going to show you. It'll just take 1 second. Okay, it will take me more than a second. Gee. You guys know me already. And the other reason is what we're doing is we're measuring fluorescence. We're not measuring FRET exactly. So we're inferring that there is some FRET because of fluorescence. And so I mentioned that this is an amazing statistical technique when you have tons of data. Imagine you have something like this between two states, and now add some noise to it. And this is some Gaussian noise. I mean, the question is, can you ever extract the signal from this? And the answer is, you can. I mean, the noise can be like 10 times bigger than the signal, and you still get out the signal. absolutely amazing.